Hi guys! Welcome back to the second episode of Lab on Screen.
In today's episode, we are going to extract DNA from fruits.
We are the iGEM Stockholm 2020 team. You can find out more about us and our project on this link right here. But before we start the experiment, let's figure out what DNA actually is. DNA stands for deoxyribonucleic acid. It's composed of two chains made of nucleotides. The nucleotides consist of a deoxyribose sugar with 5 carbon atoms, a phosphate group, and a nitrogenous base. There are 4 types of bases in DNA: adenine, thymine, guanine, and cytosine. Adenine pairs with thymine, while guanine pairs with cytosine. This causes the DNA molecule to form a double helix structure, which looks like a long twisted ladder. In prokaryotic organisms, like bacteria, DNA is located in the cytoplasm. While in eukaryotic organisms like animals, plants, and humans, DNA is located inside the nucleus of every cell. DNA carries genetic information that instructs cells to grow, reproduce, or perform important functions for life. DNA can be extracted, modified, and sequenced for several purposes. Here, we can extract DNA from fruits in a simple way.
So, today we will be extracting DNA from different types of fruits. What do we have here?
Strawberries and bananas.
Great! But you can use any kind of fruit you have at home, right?
Yeah, but the amount of fruit you will need to extract DNA differs.
But if you have, for example, strawberries or bananas or a kiwi, that would work great. So, before we can start the fun, I think we should go through the safety instructions for this experiment. As always, it's always good to wear gloves, even though you're not using anything dangerous here.
And then wear your lab coat or something to protect your clothes, like this.
If you are a child at home, please perform this experiment under adult supervision. And most importantly, please do not drink any of the solutions that were produced during this experiment.
So for today's experiment, we will be needing detergent. You can use any household detergent like dishwashing soap. You will need filter paper. You can use coffee filter paper, for example.
And then we need alcohol or disinfectant. And then we use salt.
We have some regular tap water. And for our fruits, we're going to be using a banana and some strawberries. We will also be needing some plastic bags.
And you will be needing some containers. We're using e-flasks here, but you can use any kind of glassware at home. And you will also be needing a spoon to stir with, and a tweezer to pick up the DNA. Okay, so let's start with the experiment. We will start with crushing the fruit. So let's start with removing the stem from the strawberry. Put it to the side here. And then we put the strawberry in our plastic bag. And then we can use one of the flasks, or if you have a spoon or whatever at home, to crush it. Now you have your crushed fruit. We can start making the solution.
And then we go to the next step. We need the water, detergent and salt. We will mix them all. Already prepared water, and then you can put like not that much of detergent inside. Okay, and then a little salt, and then mix them, or I can use spoon also. So yeah, to mix them all. Just make sure that all is soft. Okay.
So after we have prepared our soap solution, we are going to add some of it to our fruit mixture.
So to filter out the DNA into the floss, you can use a filter paper and shape it like a funnel. You want to pour the mixture?
Yes. So I will be pouring the strawberry mixture.
Perfect. And you can squeeze it out a bit so you get as much of the DNA.
But be careful not to squeeze out the strawberry bits into the solution.
So, and then we need alcohol to precipitate our DNA from the strawberry. We just pour it here. Okay, I think it's enough.
We have some white precipitate in the solution. So now we will try to remove the DNA and put it in another container with alcohol. Should we see if we can pull it out? Now you can see the precipitated DNA in our alcohol solution. So let's try this with banana. Repeat the same step where you crush the banana. Let's add some detergent solution. Perfect, and then you shake it and you mix it together.
And then we put the filter pepper into the container that we have. And the next step is pouring our solution and filter it. Now we're going to pour some alcohol over our banana solution.
Let's go through what happened during the experiment. Why do we use strawberries and bananas? Most organisms only have one set of DNA per cell, whereas strawberries contain 8 sets of DNA and bananas have 3 sets of DNA. Thus, both of them will produce a lot of DNA that we can clearly see during the experiment. By crushing the fruit, the cell breaks. In order to further break the cell, a detergent is used to lyse the cell. This releases the DNA into the solution. Detergent molecules contain a polar and nonpolar group, which bind to the cell membrane and separates them from the DNA molecule in the cell. DNA is a polar structure with a negatively charged phosphate backbone and soluble in polar solution like water. Salts contain a positive charge on the sodium ion that bind the negative charge of DNA, causing the DNA to become less hydrophilic. Salts help to collect the DNA strand so that we can see them easily. Alcohol precipitates the DNA out as alcohol has both polar and nonpolar groups. The polar groups help to dissolve into the water, while the nonpolar group protects the sodium cation and phosphate anion complex from the water. This causes the force of interaction between sodium cation and phosphate anion to increase. Alcohol pulls out the DNA by precipitation into a solid visible form.
Thank you so much for joining us in this experiment today. Hope it went well for you guys at home.
If you're doing this experiment at home, don't forget to send us some pictures of how your experiment went out to our social media accounts. And also, let us know down in the comments from which fruit you were able to extract the most DNA out of.
Looking forward to see you guys in our next episode. Bye-bye!